Hi everyone, welcome back to part 3 of the central dogma. So we have come a very long way in our journey of gene expression and a very long way in terms of our information flow. And we have finally made it to the third and final part. So we are going to talk about translation, and we're going to answer the question of how is the RNA that we use during the process of transcription, how is that used to make proteins? So we've already talked about the genetic code, so we know that codons are involved, but we're going to talk about how does this all actually happen. So we know the ribosomes are somehow involved, the tRNAs are somehow involved, and how do all of these processes come together to make proteins? So we're going to start with a brainstorm, and for this brainstorm question, we are going to think about what are the pieces that we need for translation. So we first need our ribosomes, and for our ribosomes, they're going to help us make protein. We need our template because everything in all of these processes, be it replication, transcription, or translation, requires a template. So our template is going to be our mRNA that came from transcription. And then we are going to need our tRNAs, which will help carry the amino acids. And of course, we need the amino acids, which are our building blocks. So on this page, and I will actually go ahead and link it in the description box as well, is a link to a YouTube video, and this video does a really nice job of walking us through this information flow, so walking us through the process of transcription and translation, and instead of looking at my really horrible drawings, you get to look at really nice visual representations of these processes. So let's spend a little bit more time talking about some of these key players. So we've talked a little bit about our mRNA, so we know all about the template, and we know how to read that, and we know how to translate it because we are pretty close to experts with regards to codons at this point, right? And we know amino acids, so we had an entire lecture topic earlier in the semester about amino acids and proteins. So we're going to focus on ribosomes and tRNAs because these are pretty new to us. So we did talk about ribosomes earlier in the semester, and when we talked about the ribosomes earlier in the semester, we pretty much just said that they have a large subunit, a small subunit, and they help with the synthesis of proteins. So today we're going to spend a little bit more time on that. So we're going to draw our spaceships again. So recall that ribosomes have a large subunit, as we just said, and then a small subunit. So they kind of look like these little spaceships, and it's probably way back in a neuron somewhere that hasn't been used in a while. But if you recall, there are also differences in the sizes of these subunits between bacteria and eukaryotes. So bacteria for their large subunit has a 50S size and a 30S for the small subunit, and together the whole ribosome is 70S. And students always ask, why don't they add up? And part of the reason that these don't add up is the S is a Svedberg unit. And when we first identified ribosomes and started looking at the pieces of them, we separated everything using a gradient density. And so the S is just a specific type of unit. And so unfortunately, when the ribosomes are in their pieces, they have different density rates than they do when they are together, which is why they don't add up. Now, when we compare bacterial ribosomes to eukaryotic ribosomes are larger. So their large subunit is a 60S and their small subunit is 40S. Together, they make up an 80S size ribosome. So the key here is that eukaryotic ribosomes are a little bit larger than bacterial ribosomes. And it actually turns out that because of these slight differences in sizes, many of our antibiotics that we use against bacteria, they target the bacterial ribosomes, Unfortunately, because the overall structure of the ribosome, even though there are slight size differences, is very similar, whenever we use antibiotics that target ribosomes, there actually happens to be more side effects that we as humans experience, tying back again to this idea that ribosomes are similar, even though their sizes are a little bit different across different domains of life. So let's go ahead and let's take a closer look inside of a ribosome. So I'm going to draw kind of like a fatter ribosome here, just so I can draw all of the pieces. So here's our large and our small subunit. We're going to draw a piece of mRNA going through it. So kind of looks like an uneven sandwich, and that mRNA sits right between the small and the large subunit. And then within that large subunit, we're going to draw kind of 3 structures here. And these 3 rectangles that we drew represent the 3 sites that are in a ribosome. And we call these sites the EPA sites. So E stands for exit, P stands for peptidyl, and then A stands for aminoacyl. And right now, these won't mean much to you, and that is okay. We are still building, and when we get to actually drawing the process of translation, you will see how these sites come into play and how they are used by both the ribosome and the RNA and, of course, our tRNA. So the importance of these sites will become a little bit more clear as we continue. So for now, just bear with me as we are building. So let's now talk about our tRNAs. So for our tRNAs, their entire job is to carry amino acids. And on the right here, we have 2 models of tRNAs. So both of these are just pictures of what they look like. So here is a folded up tRNA. Here's kind of more of a flattened version. Now, when I draw tRNAs, I tend to draw the flattened version. So this is how I draw a tRNA. A really poorly drawn clover-like molecule is really what it comes down to. So when it comes to these tRNAs, again, they are going to carry the amino acids. So what I'm going to do is draw a tRNA, and then I'm going to attach an amino acid here. So the one that it is not necessarily important. And let's go ahead and draw an mRNA. So here's our mRNA. And let's draw a codon. So here's an AUG, which hopefully we all recognize as a start codon. And you can probably start to guess how this tRNA will interact with this mRNA. So let's go ahead and remind ourselves, this is the codon. Well, everything so far in replication, in transcription, and it's true in translation as well. We think back to our rules. Everything so far has dealt with things being anti-parallel and things being complementary. So we can probably start to hypothesize, well, if we have an mRNA that is 5' at this end and 3' at this end, we know that tRNAs are RNA, which means they have to have 5' and 3' ends because all nucleic acids have 5' and 3' ends. Well, we're smart enough to figure out this end up here. If you imagine taking this part that's folded over and flattening it out straight here, this has to be a 3' end. And we know that again because we have a 5' end here. And so anti-parallelism is a common theme throughout the entire process of the central dogma. So that tells us this end right here where that amino acid is stuck, this is a 5' end. So the other thing that we can go ahead and think about is our second rule that we wrote up here, and that is complementation. So everything is complemented in these processes. So if we have an AUG, well, an A so let's start right here, can only base pair with a T, and that's in DNA, but we're talking about RNA, which has U's. So we're going to have a U that base pairs, a U base pairs with A, and a G base pairs with C. And so these will hydrogen bond, which is another common theme that we see. And this part of the tRNA that interacts with the codon is called the anticodon. And the anticodon is important for two things. Number one, it is important because this is how we make sure that we are reading the codon properly and the right amino acid gets attached. And number two, we're going to talk about how is an amino acid attached to a tRNA, and the anticodon plays a role in that as well. So we can add two things to our rules here, and that is that a tRNA, we tend to want to write them from 3' to 5', and they have an anticodon, whereas our codon is written 5' to 3'. So that anticodon is going to be really important to make sure that the correct amino acid comes to the ribosome, and we do this by hydrogen bonding to the codon. And if it's not the right one, it's not going to hydrogen bond properly, so it's kind of like a good check, if you will, for the cell. And the other thing that the anticodon is important for is attaching an amino acid. So how is an amino acid attached to a tRNA? And we call this charging a tRNA. So what's going to happen is we are going to start with a tRNA, and this is going to be an empty tRNA. So let's add our anticodon on here, and we're going to have an amino acid, and this is going to require 1 ATP because we are adding things on, and we have a very special enzyme that does this, and this enzyme is called an amino acyl tRNA synthetase. So that amino acyl site that we talked about comes back to this idea of an amino acyl tRNA is one that has been charged and is carrying an amino acid. So you can probably start to guess that that A site is where these tRNAs carrying amino acids are going to come into. But the whole job of this amino acyl tRNA synthetase is it is going to read the anticodon, and it's going to attach the correct amino acid. So this anticodon is important for two things: the charging of the tRNA by the amino acyl tRNA synthetase, and to double check that we have the correct amino acid via hydrogen bonding between the anticodon and the codon. So at the very end, after this enzyme is used, now we have a beautifully charged tRNA, and that tRNA is now ready to go and to be used in translation. All right, so just like we did with transcription, let's do a general overview of translation. And you'll notice that the steps are the same in terms of the names. So just like we had in transcription, we're going to have initiation. So we're going to have things called initiation factors. And the first tRNA is going to recognize the start codon, so that AUG, and then during initiation, we're going to assemble our ribosome. For this next step, we're going to have elongation. So for transcription, we had elongation of the RNA, and here we're going to have elongation of our polypeptide. So the ribosome is going to read the codons, and the protein is synthesized. And then last but not least, just like in transcription, we have termination. And when we reach the stop codon, we're going to have release factors bind. And essentially what they do is they stop translation. And they do that by popping off the ribosome from the mRNA. So those two pieces come apart, that releases the ribosome, releases the RNA, and releases our protein. So what we're going to do is we are going to go through each of these stages and we're going to draw what happens. So let's start with initiation. So for initiation, we're going to have our mRNA, and the first thing that's going to happen is our small ribosome binds, and what it's going to do is it's going to look for the AUG, and it's kind of cool because it scans along the mRNA until it finds the AUG, and then our initiation factors bind. So we'll make our initiation factors these little black circles. And the next thing that has to happen is our first tRNA, which we know carries methionine, binds. And the reason we know it carries methionine at first is because we know that after using the codon chart that AUG codes for methionine. So let's go ahead and draw our tRNA carrying met here. It's anticodon, so we can actually draw our 5' and 3' ends. So when all of these things assemble, we have our first tRNA, our small ribosome has found the AUG, our initiation factors bind. This now is going to cause the large subunit to bind, and when this binds, we have now a fully assembled ribosome, and when this occurs, our initiation factors are released, and then now we are ready for translation. And we can actually go ahead and write up our sites. So we have the E, the P, and the A. So when the ribosome is fully assembled, that first tRNA carrying the methionine sits in the P site or the peptidyl site. And we will see here in a second why that is important. So let's start with a fresh page. And now we're going to zoom in on the ribosome and we're going to talk about elongation. So because of how many moving parts there are in the ribosome, I'm actually not going to draw the large and the small subunit together. But in this picture, they should be drawn together, but we're just simplifying it. So what we're going to do is focus just on the key parts. So we're going to draw the mRNA. So here's our 5' to 3' mRNA, and we're going to focus on those EPA sites. So here is our first tRNA. So this is the one that we drew on the previous page. So we are picking up right here. But again, instead of drawing the large and the small subunit, I'm just focusing on the sites and the mRNA. So now that we have put together the ribosome, we're ready for translation, we're going to draw another amino acid carrying tRNA. So let's label our 5' and 3' ends. So this is going to come in to the A site. So that A site stands for amino acyl. And as that new tRNA enters, what we're going to do is we're going to double check the anticodon and codon base pair. So as that tRNA enters, we're going to, using that hydrogen bonding, make sure that the anticodon and the codon complement one another. And to do this actually requires energy. So this is going to require a molecule of GTP. So GTP is another energy management molecule, very similar to ATP, but the base is different in the nucleotide. But the principle is the same. This is simply just an energy management molecule. So it takes energy to double check things. After we have double checked that this is in fact the correct tRNA, now we have a ribosome that has 2 tRNAs in it. So one occupying the P and one occupying the A. Now what we have happening is we now actually are going to do the peptide bond formation. So this amino acid bonds with our blue amino acid. So we get peptide bond formation. So that's a blast from the past, right? It's been a while since we've heard the terminology peptide bond. So after we get formation of that peptide bond, now we have our first tRNA that no longer has an amino acid on it because that amino acid was bonded to the one on our blue tRNA. And now we have a blue tRNA that has its original amino acid and has the amino acid from the previous tRNA. So congratulations, now we have a dipeptide. How exciting. So now what we have to do is we have to have a shift, right? If the A site is where tRNAs come in, well, that A site is not empty. So the next step that happens is we actually move the ribosome down a triplet, right? And our triplet, of course, is our codon. So literally the entire ribosome shifts down. And this takes energy. And during this, we're going to use a molecule of GTP to shift that ribosome down. So after the ribosome has shifted down for our EPA site, now in our E site, we have that original black tRNA that was carrying our first amino acid. This is actually going to exit, which is why it is called the exit site. And in our second spot now, our P, now we have our blue tRNA that has our 2 amino acids on it. And now we can repeat this process. So now that we have shifted down a triplet, let's add a pink tRNA here carrying an amino acid. Now that A site, that amino acyl site is open, and now another tRNA can come in, and we repeat this cycle over and over again. And so this will continue until the stop codon is reached. So what I encourage you to do is to draw this process out and to walk yourselves through it. And again, I think that the 3D video will help you see how these pieces are moving as well. So let's talk about termination. So we're going to continue this process of a tRNA entering at the amino acyl site, a peptide bond being formed, and then the ribosome shifting, and now we have a empty tRNA that can leave via the exit site, and this happens over and over again until a stop codon is released. So at this point, when we reach a stop codon, I'm going to go ahead and just write stop because, of course, there are many stop codons. So what we're going to have is the ribosome will have, of course, shifted, and we're going to have a really long protein probably at this point. So I'm just going to draw a protein with a couple of amino acids here. And it turns out when we reach the stop codon, there is no tRNA that matches up with it. And so what we have instead is we have a release factor. And this release factor is a protein. And the release factor is going to essentially pop open the ribosome. And so after the release factor binds, we get our mRNA that is released, and we get the large subunit and the small subunit of the ribosome. And then, of course, we get our protein. So again, termination just requires a release factor. Just like we have initiation factors that are proteins that help us initiate, we have release factors that help us terminate the translation. Too bad they didn't call them termination factors, but nevertheless, I digress. All right, so now we have successfully covered all of the parts of transcription and translation. So I really like this picture because I think it does a really nice job of showing us how all of the pieces come together. So we start here with DNA, and of course, believe it or not, here's our coding strand, here's our template strand. So I hope that the practice problems are helping you all with the concept of coding and template. Here's our mRNA that we made first using DNA as a template, and then here are our tRNAs that are carrying our amino acids that match up here with the codons, so those anticodons and the codons that are hydrogen bonding to one another. So what we're going to do is we're going to do a really fun challenge here. And I think this is one of the most amazing principles when it comes to transcription and translation, and that is that I can give you a tRNA anticodon, or I can give you a codon, or I can give you a piece of DNA, be it coding or template or mRNA, and you can figure out all of the other pieces. So what I encourage you to do is, here is an anticodon. So from 5' to 3' we have A, G, C. Using this tRNA anticodon, see if you can figure out the codon, the amino acid that the tRNA is carrying, the DNA coding strand sequence, and the DNA template strand sequence. All right, so there's a couple different ways that you can start this problem. Whenever I am presented with an anticodon, what I like to do is I like to make sure that my anticodon is rewritten from 3' to 5'. And the reason for that goes back to our mRNA. So our mRNA is read from 5' to 3', and because our codon table goes 5' to 3' and everything is anti-parallel and complementary, I like to go ahead and just rewrite that for myself. So what I'm going to do is, for the anticodon, I'm going to rewrite it from 3' to 5'. So reading from 3 to 5 going backwards, that gets us to C, G, A. And now that we have our anticodon written, we can actually go ahead and figure out our codon. And there's a lot of different ways you can do this. You can draw a picture if you would like. So 3' to 5', C, G, A. So here's our mRNA. So for mRNA, we would have a G, C, U. So our codon from 5' to 3' is G, C, U. We can use that codon and our codon table to figure out what amino acid this is going to be. The amino acid is alanine, so we can write ALA here and have that tRNA carrying an alanine. But now that we have our codon, which of course is our mRNA sequence, we can figure out the DNA part. So if you recall thinking back to DNA, for mRNA, the strand of DNA that is mRNA-like is our coding strand. So our coding DNA is going to be just like our mRNA, except where there is a U, there's going to be a T because DNA has T instead of U. So our coding DNA strand is GCT. Now our template, we can use our DNA to figure that out using the idea of complementation and anti-parallelism. So G base pairs with C, C base pairs with G, and T with A. So just from one tRNA anticodon, we are able to figure out all of the other pieces that we need. So again, all of these types of problems are on Moodle. There is, I believe, 2 to 3 pages worth of them. I think there's 6 problems in total, but there are a lot of pieces to them, and I've also posted an answer key, so I highly encourage you to go ahead and take a peek at those and work through those practice problems. So the very last thing that we're going to do is we're going to compare and contrast the central dogma in bacteria and in eukaryotes. So we've talked a little bit about some of the differences, but let's go ahead and kind of have a page where we highlight those similarities and those differences. So in terms of similarities, both bacteria and eukaryotes do transcription and they do translation. They will both use RNA polymerase. They will both use ribosomes. So we're going to focus on the key differences here. So for bacteria, for transcription and translation, both of them will occur in the cytoplasm. And this, of course, goes back to our cell structure. So for bacteria, they do not have a nucleus. And so everything that they need to do transcription and translation is all in one place. The other key difference here is that no RNA processing occurs. And going back for a second with transcription and translation happening in the cytoplasm, I think probably the coolest thing about bacteria when it comes to the central dogma is they can start translation before transcription is even finished. And because everything is just kind of floating around in the cytoplasm, as long as there is enough mRNA, even if it's not fully finished being transcribed, that ribosome and those, there are different initiation factors, but they can find the start site and will start translation. So you can get this really cool thing in bacteria where you are transcribing a gene, but you're also starting to translate it right away. And it's just absolutely amazing. And that is something that cannot happen in eukaryotes. And the reason for that is these processes happen in different locations. So transcription is going to happen in the nucleus. And because we have transcription in the nucleus, and then we have translation in the cytoplasm, we have extra work we have to do. We have to do RNA processing. So that RNA processing, of course, is the cap and the tail, and then splicing. So we put the cap and the tail on for protection from degradation. And part of that, again, is because we have to go from the nucleus into the cytoplasm. And we don't want that RNA that we just spent so much time making, we don't want that to be degraded. So last but not least, here is just a picture highlighting some of those differences. So here we've got DNA in bacteria being transcribed to mRNA and translated to protein, and it all happens in the same area, so all of it happening in the cytoplasm. Here in the eukaryote, we've got our DNA that is made into pre-mRNA, and that mRNA is then processed into mature mRNA with a cap, a tail, and splicing, and then it has to be exported into the cytoplasm, and then of course we get translation. So congratulations, you have survived the central dogma, which I think is just one of the coolest topics in the entire world, and to this day it still amazes me. I hope you all have enjoyed this topic as much as I have, and when we come back for our last topic of the semester, we're going to talk a little bit about mutations and how mutations fit into the central dogma, and we're going to go through a really cool case study. So thanks everyone, have a wonderful rest of your day, and I'll catch you in the next one.